Now that you know a bit about gene expression, let's look at what a microarray is and how it can be used to compare gene expression in cells. A DNA microarray uses a glass chip that contains multiple wells. Each well has multiple copies of the same single-stranded DNA for a single gene. However, each well on the array contains a different gene than the wells next to it. With chips that contain thousands of wells, scientists can use DNA microarrays to measure the expression levels of large numbers of genes simultaneously. So how do scientists know whether the genes in these wells are being expressed? It all starts with the cell's mRNA. In part 1, you learned that if a gene is being expressed, the cell is making mRNA. The more the gene is upregulated, the more mRNA is made. If the gene is downregulated or not being expressed, then little to no mRNA is produced. Because mRNA is an indicator of gene expression, the first step to perform a microarray is to isolate mRNA from the cells of interest. In this case, these cells will be from healthy and cancerous tissue. After mRNA is isolated, the mRNA is reverse transcribed, a process in which an enzyme transcribes the mRNA into DNA to create complementary DNA or cDNA. During the reverse transcription process, complementary DNA is labeled so it can be visualized in the microarray. In this case, normal cDNA is labeled with a green fluorescent dye, and cancer cDNA is labeled with a red fluorescent dye. After labeling, the red and green cDNA are both applied to the same microarray. As the cDNA washes over the chip, it will hybridize or bind to any well that has its complementary strand. Remember, each well contains single-stranded DNA from a specific gene. Therefore, if the healthy cells had those genes turned on, they made mRNA, which was reverse transcribed to the cDNA. That cDNA hybridizes to the DNA in the well of the gene it represents. The same is true for the cDNA from the cancerous tissue. Now the microarray is ready to analyze. To analyze the microarray, it is placed into a scanner that can read the fluorescent labels. The green labels are scanned and an image is produced, followed by a scan for the red labels. After both the healthy and cancerous cDNA has been scanned, the two images are overlapped to more easily visualize the similarities and differences between gene expression. Spots that are red represent genes that are more strongly expressed or more expressed in cancer cells, whereas spots that are green represent genes that are more strongly or only expressed in healthy cells. Yellow spots indicate the gene is expressed equally in both cancer and healthy cells. Spots that are black or gray indicate the gene is not expressed in either cell type. In this array, the spot for the gene CDK4, cyclin-dependent kinase 4, is located here. This gene creates a tumor suppressor protein. Because the spot for it is green, we see the protein is only produced in healthy cells. This means that in cancer cells, the gene is not expressed. Therefore, the cell no longer produces this protein to stop tumors from growing. Because each spot on the microarray is associated with one gene, multiple genes can be compared at one time to see which ones need to be studied further, as they may contribute to the development of cancer. Scientists would most likely want to study genes that show up as red or green, because these show a difference between the expression of that gene in healthy versus cancerous cells. Because the yellow spots show an equal amount of expression in healthy and cancerous genes, these genes most likely would not be picked for additional study. Also not of interest would be the black spots that show no expression in either cell type. After genes of interest are identified from the microarray, The DNA of those genes can be sequenced in both healthy and cancer cells to find the mutation. This tells us exactly how the protein has been altered and allows us to then look for new approaches to treatment. A DNA microarray is useful in so many genomic applications that it has become an essential tool. Aside from studying cancer, microarrays help researchers study how patients might respond to a particular drug. In toxicological research, microarrays can help scientists track how the exposure to certain toxins can change the genetic profile of an organism. Microarrays are also a useful tool in clinical microbiology, as they can help identify bacteria and pathogens. With more than a dozen other ways microarrays can be used to study genes, the vast information gathered from them will help change the field of genomics as we know it.